Welcome everyone. In the previous video we were discussing the different changes which takes place in the zygote after fertilization. That is, zygote will develop into an embryo. How the development is taking place, let us see this one. So we have already started with this. So this is the zygote, this diploid zygote which is in diploid condition. Okay, it has undergone mitotic division and produced two cells. So these two cells, one is towards the micropylar end and the other towards the callosal end. The cell which is towards the callosal end, that is called as the terminal cell, and the cell which is towards the micropylar end, this is called as the basal cell. We have also seen this terminal cell has divided and given rise to proembryo. Terminal cell has divided and given rise to proembryo. This one, this is the upper one. So it is this blue colored one. This is the cells which has formed because of the division of this terminal cell. Terminal cell has undergone mitotic division and produced 4 cells. Okay. So two divisions, it has divided twice. So repeat after the mitotic divisions, the terminal cell gave rise to this part, and after the repeated divisions of this basal cell, we have got the suspensor and this haustorial cell. Haustorial cell is not different, it is just a part of suspensor itself, but the down, that is the last cell of the suspensor, it is a little bigger. That is why the name is given the haustorial cell. Okay. So now the cells which have formed from this terminal cell, the cells which has formed from this terminal cell, so this one is called as the embryo. This mass of cells, which has formed by the division of this terminal cell. So this mass of cell is actually the embryo. We call it as proembryo. So as we have this 4 cells, that is the mass of cells are 4 in number. So we call it as proembryonal quadrant. So first, before going to this we see this type of development is seen in dicots. Okay. So the development, this is the development of dicot embryo. What we are saying is the development of dicot embryo. Okay. How embryo is formed in dicotyledonous plants or dicot plants. So here this proembryonal quadrant has been formed because of the divisions of this terminal cell, and suspensor is formed because of the divisions in the basal cell. Okay. Now what is the function of the suspensor? It pushes, and this haustorial cell, it pushes this proembryo towards the endosperm. For example, if I have drawn this is the ovule. Okay, here this is the micropyle, here we have this embryo sac, So inside this embryo sac we have this proembryo. Okay, inside this, if I draw this one, this is the proembryo and this is the suspensor. Because development is taking place at the micropylar end. We have already discussed this in the previous video. So this is the haustorial cell, bigger cell. So and here in this one we have this endosperm, that is nutritive tissue. This whole embryo sac is filled with this nutritive tissue. The synergids and antipodals have been degenerated. The only thing which we find is the endosperm and the that is zygote, which is changed into embryo. So this suspensor, this haustorial cell and this suspensor will push this embryo towards the endosperm, so that the embryo gets the nutrients. So that is the major function of this suspensor. It is pushing the embryo. This is the main embryo. The suspensor and this osteorial cell is pushing the embryo towards the endosperm. Okay. So by taking the nutrition from the endosperm, this proembryo will further divide. This proembryo will further divide. Okay. And it produces more mass of cell. So first it was 4 cells. For example, the mass of cells were 4. Okay. Now here this 4 cells will again divide and produce 8 cells. 4 cells will again divide and produces 8 cells. 1, 2, 3, 4, 5, 6, 7, 8. Actually it won't be like this because it is just mass of cell. 8 cells will be there, but it is not like all will not be seen. So if we see this mass of cells means some will be seen from the front, some will be at the back. It won't be like this. Just for the understanding, I am drawing this like this. So this one. So this is this proembryonal quadrant has further divided and produced mass of cell. So this is called as the proembryonal octet because the mass of cells, number of cells here is the 8. So proembryonal octet has been formed. Okay. Now this the cell which was near this, the suspensor cell which was near this proembryo, this will change into hypophysis. This will change into hypophysis. Okay. And the rest of the cells are the same. The cell which is near to this proembryo, the cell which is near to this proembryo, this will change into hypophysis. This will change into hypophysis. This is the suspensor and all. Okay. Now this one, this is the proembryonal octet. Now this proembryonal octet will further undergo divisions, periclinal divisions. So now if I see this, if there is divisions, so mass of cells will be produced, So it is was octet now, 1, 2, so octet was there. Now it undergoes further division and produces more mass of cells. Okay. But the division here is periclinal. The division here is periclinal division. That is, one cell will be towards outside and one cell will be towards inside. In periclinal division, how this will be seen? Okay. This is the octet now, embryonal octet. It will divide and produce mass of cell. It will divide and produce mass of cell. So here in this now it will divide and produce another cell. So this will become the inner cell and this will become outer cell. So it will also divide and produce one more cell. So this will become inner cell and this is the outer cell. So this will divide and produce one more cell. So now let us, I am not saying for the understanding I am drawing. So for example, this is the cell, this is one cell, it has undergone division and produced another cell. So one will be the outer cell, one will be the inner cell. Okay. So this has undergone. So like this what we get is the one outer cell and one inner cell. One outer cell and one inner cell will be getting. So this is the where this proembryonal octet, it undergoes periclinal division and each cell will divide and produce one more cell. So one will be towards the outside and one will be towards the inside. Okay. It won't be resembling like this. It won't be resembling like this, just for the understanding, because here how the cells, how these cells, what will be the fate of these cells, what these cells will form, these cells will change into which organ. Okay. So we will see this, that is why we are drawing it like this one. So it undergoes periclinal division, it undergoes periclinal division. So now this embryo is called as globular embryo. The first stage is the zygote. Zygote has changed into proembryonal quadrant. This proembryonal quadrant will further undergo divisions and changes into proembryonal octet. Okay. Now here this hypophysis is formed and the suspensor and all is the same. Here in this proembryo, in this proembryonal octet, periclinal division will take place, and because of this periclinal division the cells will be arranged in such a manner that one will be towards outside and one will be towards inside. Okay. Now let us see this. We have said this hypophysis has been formed. So this is because of some reason the cell which was the nearest to this proembryo, it has changed into hypophysis. This when this periclinal division will take place, along with this periclinal division, here in hypophysis also repeated division will take place. This was one cell. Okay. This one cell has been repeatedly divided and produced a mass of cell. This hypophysis has divided and produced mass of cell, and this proembryonal octet, it also has divided and produced mass of cell. So this is periclinal division, and here transverse division is taking place. So mass of cell is formed. So now let us see what gives us what. So from this if we see this hypophysis, this two layer of cells, so it will change into root tip and root cap. This one, these cells will change into root tip and root cap, and these two cells will change into root cortex. Root cortex is the tissue which is present inside. What are the different tissues which are present inside the root? Different tissues which are present inside the root. So these two, that is few cells of this hypophysis, it will change into root cap and root tip, and the cells which are near this proembryo, the cells of hypophysis which are near this proembryo, this will change into root cortex. This will change into root cortex, and this is, so this one in this that is in this proembryo, half of the proembryonal cells will change into hypocotyle. Okay. For example, this half will change into hypocotyle, and this half will be changed into plumule and cotyledons. This half will change into plumule and cotyledons. This half is changed into, it will change into hypocotyle. This one, the cells which are that is hypophysis cells which are near this proembryo, this will change to root cortex, and the hypophysis cells which are towards which are away from this one proembryo, which are towards the suspensor. Okay, these cells, these hypophysis cells, it will change into root cap and root tip. How it will change? Okay, let us see this one. So here this will change into, these cells will change into root cap and root tip. Okay, so this will change into root cortex. I will draw it like this. It will further divide and change into, mass of cells will be formed. It will be changing into root cap and root tip. Okay, these cells will undergo rapid divisions and form the that is root cortex, the tissue which is inside this tissue which is inside the root. Now here also many divisions will take place, mass of cells will be formed. Many number of cells will be formed, and this will change into hypocotyle. Okay, and here also mass of cells will be formed. So here from this part we get the plumule. From this part we get the plumule, and from this part, so this part will change into one cotyledon, and this part will change into another cotyledon. This part will change into This part will change into one cotyledon, this part will change into another cotyledon. So it resembles to that of heart shape. It resembles to that of the heart shape. So like this, it resembles like that of heart shape. So now this embryo is heart shaped embryo. So this was, earlier it was globular embryo. This globular embryo has changed into heart shaped embryo. Globular embryo after repeated mitotic divisions in all the cells, so it has changed into a heart shaped embryo. So first embryonal, the zygote undergone division, embryonal pro-embryo is formed. Okay, from the zygote pro-embryo quadrant is formed, and from pro-embryo quadrant pro-embryo octet is formed. Pro-embryo octet has changed into globular embryo, and this globular embryo after repeated mitotic divisions has changed into heart shaped embryo. Has changed into heart shaped embryo. Okay. So now let us see what are the different changes which takes place. So, but here what happens is, let us say this, all this, where it is happening, all this is happening in this embryo sac. All this changes is happening in this embryo sac. So, embryo sac is a very small structure. The cotyledons are growing. Okay. So, what happens is, they will show bending. This cotyledons will, see here, the suspensor is continuously pushing towards the endosperm. So, till this embryo is developed, sometimes what happens, whole endosperm will be utilized by the embryo. Sometimes when this embryo is developing, it utilizes this whole endosperm. So, there won't be any more endosperm. Okay. So, such seeds which does not have any endosperm remaining, because I said you the ovule will change into seed. So, such seeds where we do not find this endosperm, now only embryo is there. So, this embryo, this developed bigger embryo will be there in the embryo sac. It has utilized almost all the endosperm. So, now the space is very little in this one. The space is very little in this embryo sac. So, what happens? These cotyledons will show bending. The cotyledons will show bending. This is one cotyledon and this is another cotyledon. As the size is very small, these cotyledons will show bending. They will bend towards one side. They will bend towards one side. Plumule will be as it is here. This has grown. So, now it does not have space now. It tends to bend at this side. So, this is also growing. It tends to bend at one side. So, if I draw this one, this is, see here, this is one cotyledon and this is another cotyledon. Okay. So, I said you this region will change into cotyledon and plumule. So, plumule is already there here, plumule, this is the plumule, these are the two cotyledons, and this is the, this region is the hypophysis, the hypocotyle, this is the hypocotyle, and here this is the root cortex and this is the root tip. So, this whole structure of hypophysis has changed, it will change into root. Okay. This whole structure, because this is the tip of the root and root cap, and inside the root tissue will be there, that is the root cortex. Okay. So, this whole will change into a root and these are the two cotyledons. Now, the heart shaped embryo will change into horseshoe shaped embryo. Horseshoe shaped embryo. So, here where will all this present? Inside this one. Okay. Now, this one, this ovule will change into seed, and this ovary, inside this ovary will be there now. This ovary will change into fruit. Ovary will change into fruit, ovule will change into seed, and inside the seed, this embryo will be present. Okay. Inside the seed. So, here inside the seed, because this is the ovule which is changing into seed, inside the seed that is embryo will be there. So, inside the seed embryo sac is there now, inside the embryo sac we have that is now growing embryo. So, inside the seed we have the embryo, inside the seed we have embryo. So, this is how the zygote has developed into an embryo inside, and from this embryo now the new plant will be arising. From the embryo the new plant will be arising. So, this is how from a single zygote a new plant is arising. This is how from a embryo a new plant will be arising. So, here if we see this one, so if we see the structure of seed like this, okay, so this we have seen in the pea seeds, you just open it, you will find like this, okay. So, we will see what are the different parts of the seeds in the later on, but this embryo will be inside the seed, this embryo will be inside the seed. Ovule has changed into seed, and inside the ovule embryo sac was there where embryo is there. So, inside the seed we find the embryo. So, this is how the zygote, which was the single cell, has divided and produced two cells, that is terminal cell and the basal cell. Terminal cell has given rise to proembryo and basal cell has given rise to suspensor. The suspensor function is to push this embryo towards the nutrition, towards the endosperm. That is the only function. Okay. Now, this proembryonal quadrant will further undergo changes and change into proembryonal octet, and here the cell which was near this proembryo, the suspensor cell which was near, which was at the neck, which was near the proembryo, it will change into hypophysis. So, this hypophysis and this proembryonal octet will undergo further divisions and it will form, this hypophysis will form the root radical part, and in this proembryo half of the part will form the hypocotyle, half of the part will form cotyledon and the plumule. So, these are all inside. So, this is actually the embryo which will be inside the seed, which will be inside the seed. So, we will see the different parts of the seed in the next video, but before that we have seen this dicot embryo, how zygote has changed into embryo. This is the dicot embryo. In the next class we will see how monocot embryo is formed. There are not much differences in between dicot and monocot, but still very little difference is there, but most of the things will be the same in both the embryos. So, in the next class we will see the development in the monocot embryo. Then afterwards we will see the seed, structure of seed, both in the dicot as well as in the monocot. How the structure of seed will be there in dicot and monocot, we will see it first. We will see this, how the embryo is formed in the monocot. We have seen this is how the embryo, how zygote changes into embryo in the dicots. We will see in the monocots next class.